In this lecture, we are going to discuss the difference between the three most important bacteria. First one is the Klebsiella pneumoniae, second is the Enterobacter cloaca, and the third is the Serratia marcescens. We will see how these bacteria differ from each other, and along with that we will also see the clinical scenario related to the each bacteria. So let's get started. First one is the KP, Klebsiella pneumoniae. It's a gram-negative bacillus. All these bacteria, Klebsiella pneumoniae, Enterobacter cloaca, and the Serratia marcescens, all these bacteria are gram-negative. Second, Klebsiella pneumoniae is non-motile, while Enterobacter cloaca and the Serratia marcescens are motile. So that is the important thing. Uh, so it means that we can differentiate Serratia and the Enterobacter from the Klebsiella on the basis of motility test. So that is an important thing that can differentiate the KP from the Enterobacter and the Serratia. Next is the Klebsiella pneumoniae, having prominent capsule that act as antiphagocytic, because our immune system cannot tackle those bacteria having capsule, while the KP having prominent and large capsules present on its surface. So it will prevent this bacteria from our immune system, while Enterobacter having weak capsule and Serratia having no capsule. So that is an important thing, because KP having prominent capsule, so it will produce mucoid colonies on the Meconic agar, while Serratia will not produce as such mucoid colonies on the Meconic agar. KP is a strong lactose fermenter. It means that its colonies will be pink in color on the Meconic agar, while Enterobacter is slow lactose fermenter, while Serratia is non-lactose fermenter, its colonies will be colorless. So again, on the basis of the lactose fermenter, we can distinguish KP, Enterobacter and the Serratia. Next, KP will not produce any pigment. Enterobacter will also not produce any pigment, but Serratia will produce the pigment that is red in color, and that pigment is called the prodigiosins. So that is an important thing. So it means that Serratia colonies will be red in color due to this pigment. So that is important thing, while there will be the no pigment production in case of KP and the Enterobacter. Next, if we move, so what type of colonies will you see in the KP? You will see the large mucoid and the pink colonies on the Meconic agar, because it's an important thing. On the Meconic agar, you will see mostly gram-negative growth of the gram-negative bacteria. Pink colonies, it means that it will ferment. This bacteria will ferment the lactose that is present in the Meconic agar. So it will look pink in color. So in Enterobacter case, you will also see the pink colonies and less mucoid. The KP will produce more mucoid and Enterobacter will produce less mucoid. Why? Because KP having prominent capsule, while its capsule is weak. So that's why its colonies will also be less mucoid in that case. While Serratia colonies will be colorless, because it's a non-lactose fermenter. It doesn't ferment any lactose molecules present in the Meconic agar, while the colonies will be red on the nutrient agar. That is an important thing that we have already discussed, why Serratia produce the red colonies on the agar, because it's produced red pigment in that case. Next, we can also distinguish these three bacteria on the basis of the biochemical. citrate will be positive in case of Kp. In case of Enterobacter, citrate and the motility test will be positive. So, if I want to distinguish between the Kp and the Enterobacter, so citrate will be positive in both cases. But how can I distinguish these two bacteria on the basis of the biochemical test? And that will be the motility test, because Enterobacter motility test will be positive while the Kp motility test will be negative in that case. Motility test will also be positive in the Serratia, and DNase and gelatin liquefaction test will also be positive in the Serratia in that case. So that is an important thing. If we look at the pathogenesis, a thick polysaccharide capsule present that will act as antiphagocytic, and it will also having endotoxin, because gram-negative bacteria having endotoxin on its surface. So Enterobacter having adhesins that can adhere to the surface of the body, and it can also form biofilm. That is an important thing where bacteria can survive in your body for a long period by making biofilm on different body parts. Serratia will also produce different enzymes, DNase and protease, that will help in the survival of this bacteria in the body of the human, and it will also form the biofilm in that case. Next, we will see what will be the symptoms related to these bacteria. So Kp causes pneumonia, and the most important feature of this type of pneumonia and caused by the Kp is the current jelly sputum, reddish sputum in that case. And it can also cause the UTI, urinary tract infection, and the septicemia. So these are the different things caused by the Kp. You can also see the Enterobacter, Nosocomial UTI, urinary tract infections, bacteremia, bacteria in the blood, so and the wound infections. And Serratia causes hospital acquired UTI. So UTI is common in all of them. So that is an important thing. Respiratory infection and catheter associated infections also seen in this Serratia. So these are mostly hospital acquired. These bacteria, a patient can get these bacteria from the hospital. So basically these are hospital acquired infection in that case. Next, we will see clinical scenario. An elderly diabetic alcoholic with lobar pneumonia and current jelly sputum. So it's an important thing. Kp is common in the immunocompromised patient. So in the clinical scenario, you have already seen diabetic and alcoholic patient. Next, you will see culture shows mucoid. Whenever you see the word mucoid, it goes towards the Kp. Why Kp produces the mucoid colonies? Because it has thick and the prominent capsule on its surface. And you can also see the non-motile. Kp is non-motile. And lactose fermenting gram-negative bacillus, so it goes towards the Kp. Next clinical scenario is the catheterized hospitalized patient develops nosocomial UTI, urinary tract infection. So history will help you in making good diagnosis related to these bacteria, because it's a hospitalized nosocomial UTI. So in our history, patient is also hospitalized. Culture shows motile. Enterobacter is motility test is positive. Late lactose fermenting gram-negative bacillus with AMPC resistance. So these features help in finding what type of bacteria. In that case, you will see the Enterobacter cloaca. Next is this Serratia Marcescens scenario. An ICU patient with device associated infections like catheterizations is very much important, because it will get chanced in the catheterized patient to get inside the body of that patient and causes the infections, nosocomial infections. Culture shows red pigmented, because it's produced a red pigment, as we have already seen. DNA is positive and non-lactose fermenting gram-negative bacillus, because it will not ferment any lactose present in the Meconic agar. So this is how we differentiate bacteria on the basis of their properties and the clinical manifestation and a little bit history of the patient. So this is all about three most important bacteria: Klebsiella pneumoniae, Enterobacter cloaca, and the Serratia marcescens. If you still have any question, you may ask in the comment section. Thank you so much.